Chapter 16, Blood. In this section, we'll explore the composition and functions of blood, focusing on several key topics. We begin with plasma and the cellular elements of blood, examining the liquid and cellular components that make up this vital fluid. Next, we'll discuss blood cell production, exploring how and where these cells are formed. We'll then take a closer look at red blood cells, understanding their structure, function, and crucial role in oxygen transport. Following that, we'll explore platelets and their essential function in preventing blood loss. Finally, we'll examine hemostasis and coagulation, uncovering the processes that stop bleeding and maintain vascular integrity. By the end of this chapter, you'll gain a comprehensive understanding of how blood functions to support life. Blood is a specialized connective tissue with both cellular and liquid components. Plasma, the extracellular matrix of blood, is primarily composed of water, proteins, and dissolved substances. About 92% of plasma is water, serving as a medium for transporting various molecules. 7% consists of proteins, including albumins, globulins, fibrinogen, and transferrin, which play essential roles in maintaining blood pressure, clotting, and immune function. The remaining 1% contains ions such as sodium, potassium, chloride, hydrogen, calcium, and bicarbonate, as well as dissolved gases like oxygen and carbon dioxide. Additionally, plasma carries organic molecules, trace elements, and vitamins necessary for cellular function. Plasma shares many similarities with interstitial fluid but differs in its higher concentration of plasma proteins, which are crucial for maintaining osmotic balance and proper physiological function. Blood is composed of plasma and cellular elements, each playing a vital role in maintaining homeostasis. Plasma, the liquid component, makes up about 55% of blood and serves as a transport medium for various substances. Plasma consists primarily of water, along with dissolved ions, gases, organic molecules, and trace elements. These components are essential for maintaining fluid balance, nutrient transport, and waste removal. Organic molecules in plasma include amino acids, proteins, glucose, and lipids. Among the plasma proteins, albumins contribute to colloid osmotic pressure and act as carriers for various substances. Globulins function as clotting factors, enzymes, and antibodies, supporting immune defense and blood coagulation. Fibrinogen plays a crucial role in forming fibrin threads, which are essential for blood clotting. Additionally, transferrin is responsible for iron transport, ensuring proper red blood cell production. Plasma also contains dissolved gases such as oxygen and carbon dioxide, which are necessary for cellular respiration and gas exchange. Together, these components of plasma support the physiological functions that sustain life. Blood is composed of three main types of cellular elements: red blood cells, white blood cells, and platelets, each playing a critical role in maintaining health and function. Red blood cells, also known as erythrocytes, are responsible for oxygen transport. They contain hemoglobin, which binds oxygen and delivers it throughout the body while also assisting in the removal of carbon dioxide. Platelets, small cell fragments derived from megakaryocytes, are essential for blood clotting and help prevent excessive bleeding when injuries occur. White blood cells, or leukocytes, function as the body's primary defense against infection and disease. They are divided into different types, each with a specialized role. Lymphocytes, also called immunocytes, play a crucial role in adaptive immunity. Monocytes develop into macrophages, acting as phagocytes that engulf and digest pathogens. Neutrophils function as both phagocytes and granulocytes, rapidly responding to infections. Eosinophils, classified as granulocytes, are involved in allergic reactions and parasitic defense. Basophils, another type of granulocyte, release histamines during immune responses, and when found in tissues, they are referred to as mast cells. Together, these cellular elements work in harmony to ensure oxygen delivery, immune protection, and clot formation, maintaining the body's overall health and stability. Blood consists of both plasma and cellular elements, each serving essential physiological functions. The cellular elements include red blood cells, white blood cells, and platelets. Red blood cells, or erythrocytes, play a crucial role in transporting oxygen from the lungs to tissues and removing carbon dioxide for exhalation. Their unique biconcave shape allows for increased surface area, enhancing gas exchange efficiency. White blood cells, or leukocytes, are the body's primary defense against infection and disease. Lymphocytes produce specific immune responses, including antibody production and cellular immunity. Monocytes act as phagocytes, engulfing and digesting pathogens, and later develop into macrophages in tissues. Neutrophils serve as the first line of defense, responding rapidly to infections by engulfing bacteria and debris. Eosinophils specialize in combating parasites and regulating allergic reactions, while basophils release histamines and other chemicals involved in inflammatory responses. Platelets, also known as thrombocytes, are cell fragments that play a crucial role in hemostasis. They aggregate at injury sites, forming a platelet plug and releasing factors necessary for blood clotting. Together, these cellular elements maintain oxygen transport, immune defense, and clot formation, ensuring the body's overall stability and function. Blood cells are produced in the bone marrow through a process known as hematopoiesis. This process generates all types of blood cells, ensuring the body maintains proper oxygen transport, immune defense, and clotting functions. Red bone marrow is the active site of hematopoiesis, appearing red due to the presence of hemoglobin. It is responsible for producing both red and white blood cells, with approximately 25% of its production dedicated to red blood cells and 75% to white blood cells. This imbalance reflects the short lifespan of white blood cells and the body's constant need to replenish them. In contrast, yellow bone marrow is primarily composed of adipose tissue and remains inactive under normal conditions. However, in cases of extreme blood loss or physiological stress, yellow marrow can convert to red marrow to increase blood cell production. Hematopoiesis is regulated by cytokines, including interleukins and colony-stimulating factors. These signaling molecules stimulate stem cells in the bone marrow, directing them to differentiate into specific blood cell types based on the body's needs. This intricate regulatory system ensures that the circulatory and immune systems function efficiently, adapting to changes in health and environmental conditions. Bone marrow is the primary site of blood cell production, containing cells at various stages of development. This complex tissue, known as the stroma, provides structural support and a nurturing environment for hematopoiesis. The stroma consists of fibroblasts, reticular cells, collagenous fibers, and an extracellular matrix that supports stem cell differentiation. Within the bone marrow, stem cells serve as the foundation for blood cell formation, giving rise to various cell types. As these cells mature, they undergo differentiation. Reticulocytes, the precursors to red blood cells, expel their nuclei before entering circulation. Monocytes and macrophages play essential roles in immune function, while lymphocytes contribute to the adaptive immune response. Platelets originate from megakaryocytes, large bone marrow cells that fragment into smaller platelet pieces. These platelets are vital for clotting and wound healing. Mature blood cells must pass through the endothelium of venous sinuses to reach circulation, ensuring continuous replenishment of blood components. This highly regulated environment allows the bone marrow to adapt to the body's needs, increasing production in response to injury, infection, or blood loss. Through this dynamic process, bone marrow plays a crucial role in maintaining health and homeostasis. The production of blood cells is tightly regulated by specific signaling molecules that ensure proper balance and function. Colony-stimulating factors, or CSFs, play a crucial role in regulating leukophoresis, the process of white blood cell production. These factors are produced by cells within the bone marrow and stimulate the differentiation and proliferation of leukocytes, ensuring the immune system is equipped to fight infections. Thrombopoietin, or TPO, is responsible for regulating platelet production. This hormone is produced in the liver and stimulates the development of megakaryocytes, the precursor cells that give rise to platelets. Adequate platelet levels are essential for blood clotting and wound healing. Erythropoietin, or EPO, is the key regulator of erythropoiesis, the production of red blood cells. EPO is primarily produced in the kidneys in response to low oxygen levels in the blood. It signals the bone marrow to increase red blood cell production, enhancing the blood's oxygen carrying capacity. Through these regulatory mechanisms, the body maintains the appropriate levels of blood cells, adapting to physiological demands and ensuring homeostasis. Red blood cells, or erythrocytes, play a crucial role in transporting oxygen and carbon dioxide throughout the body. One key measure of red blood cell concentration is hematocrit, which represents the ratio of red blood cells to plasma, expressed as a percentage. A higher or lower hematocrit level can indicate conditions such as dehydration, anemia, or polycythemia. Unlike most other cells in the body, mature red blood cells lack a nucleus. This unique adaptation allows them to maximize space for hemoglobin, the protein responsible for oxygen transport. However, because they cannot repair themselves, red blood cells have a limited lifespan of approximately 120 days before being recycled by the spleen and liver. The morphology, or shape, of red blood cells can provide valuable diagnostic insights. Variations in size, shape, or structure can indicate underlying diseases such as sickle cell anemia or iron deficiency anemia. Another important measurement is the mean corpuscular volume, or MCV, which refers to the average size of red blood cells. Abnormal MCV values may signal deficiencies in iron, vitamin B12, or folate. Through these characteristics, red blood cells serve as key indicators of overall health and are essential for maintaining oxygen balance in the body. A complete blood count, or CBC, is a routine diagnostic test that provides essential information about the cellular components of blood. This test measures key parameters such as hematocrit, hemoglobin levels, red and white blood cell counts, and platelet levels. Hematocrit represents the percentage of blood volume occupied by red blood cells. In males, the normal range is between 40 and 54%, while in females, it ranges from 37 to 47%. Hemoglobin levels, which indicate the oxygen carrying capacity of blood, typically range from 14 to 17 grams per deciliter in males and 12 to 16 grams per deciliter in females. The red blood cell count reflects the number of circulating erythrocytes, with normal values ranging from 4.5 to 6 million cells per microliter in males and 3.8 to 5.4 million in females. The total white blood cell count, which includes all types of leukocytes, typically falls between 4,000 and 11,000 per microliter. A differential white blood cell count provides further insights into immune function by measuring the percentages of specific leukocyte types. Neutrophils make up 50 to 70% of white blood cells, followed by lymphocytes at 20 to 40%, monocytes at 2 to 8%, eosinophils at 1 to 4%, and basophils at less than 1%. Platelet counts, which assess the blood's ability to clot, typically range from 150,000 to 450,000 per microliter. Abnormalities in platelet levels can indicate clotting disorders or bleeding risks. By evaluating these values, a complete blood count helps detect a wide range of medical conditions, from anemia and infections to inflammatory disorders and blood-related diseases. Red blood cells, or erythrocytes, have a unique biconcave disc shape that is essential for their function in oxygen transport. This specialized shape increases surface area, allowing for more efficient gas exchange and flexibility as they pass through narrow capillaries. Scanning electron micrographs reveal the distinct biconcave structure of red blood cells when suspended in an isotonic medium. This shape is maintained by the cytoskeleton, a network of protein filaments that provide structural support and flexibility. The cytoskeletal framework of red blood cells consists of actin filaments and attachment proteins, which work together to maintain cell integrity. This flexible cytoskeleton allows erythrocytes to deform as they navigate through tight spaces in the circulatory system, ensuring uninterrupted blood flow. Unlike most other cells, mature red blood cells lack a nucleus and organelles, maximizing space for hemoglobin, the protein responsible for binding and transporting oxygen. However, this also means they have a limited lifespan of approximately 120 days before being broken down and recycled by the spleen and liver. The biconcave structure, combined with a well-organized cytoskeleton, enables red blood cells to efficiently transport oxygen and carbon dioxide, playing a vital role in sustaining life. Red blood cells, or erythrocytes, are highly adaptable cells that can change shape in response to their environment. The balance of solute concentrations inside and outside the cell plays a crucial role in maintaining their characteristic biconcave shape. When erythrocytes are placed in a hypertonic solution, where the external solute concentration is higher than inside the cell, water leaves the cell by osmosis. This causes the cell to shrink, but the rigid cytoskeleton remains intact, giving the surface a spiky appearance. These shrunken cells are described as crenated. In contrast, when erythrocytes are placed in a hypotonic solution, where the external solute concentration is lower than inside the cell, water enters the cell. This causes the red blood cells to swell and lose their characteristic biconcave shape. In extreme cases, excessive swelling can lead to cell lysis, where the membrane ruptures. In certain medical conditions, such as sickle cell disease, the shape of erythrocytes is permanently altered due to abnormal hemoglobin production. In this disorder, red blood cells become rigid and sickle-shaped, reducing their ability to flow through small blood vessels. This leads to blockages, pain, and impaired oxygen delivery throughout the body. These structural changes highlight the importance of maintaining proper osmotic balance and the critical role of hemoglobin in preserving red blood cell function. Hemoglobin is a vital protein responsible for oxygen transport in red blood cells. Its structure consists of 4 polypeptide chains arranged in 2 pairs, each containing a heme group. These heme groups play a crucial role in oxygen binding and release, ensuring efficient oxygen delivery throughout the body. The heme molecule is a porphyrin ring with an iron atom at its center. This iron is essential for hemoglobin function and must be continuously supplied through the diet. Once absorbed, iron is transported in the blood by a specialized protein called transferrin, which delivers it to the bone marrow for red blood cell production. Excess iron is stored in the liver in the form of ferritin, a protein that prevents toxicity while ensuring iron availability when needed. Proper regulation of iron metabolism is critical, as both iron deficiency and excess can lead to serious health issues, including anemia or iron overload disorders. Through this tightly controlled system, the body ensures a steady supply of iron for hemoglobin synthesis, supporting oxygen transport and overall physiological function. Hemoglobin is the essential protein in red blood cells responsible for oxygen transport throughout the body. Its structure consists of 4 globin protein chains, with most adult hemoglobin containing 2 alpha and 2 beta chains. Each globin chain is associated with a heme group, which plays a crucial role in binding oxygen. The heme group is a specialized structure known as a porphyrin ring. At the center of this ring is an iron atom, which is the key component that allows hemoglobin to bind oxygen. When oxygen levels are high, such as in the lungs, the iron binds oxygen molecules, forming oxyhemoglobin. As red blood cells travel to tissues with lower oxygen concentrations, hemoglobin releases the oxygen for cellular use. This iron-based binding system enables hemoglobin to efficiently transport oxygen while also playing a role in carbon dioxide transport. Proper hemoglobin function is critical for maintaining oxygen balance and supporting cellular respiration. Any disruptions in hemoglobin production or structure, such as in sickle cell disease or anemia, can significantly impact oxygen delivery and overall health. By understanding the molecular structure of hemoglobin, we gain insight into its essential role in sustaining life. Red blood cells, or erythrocytes, have a lifespan of approximately 120 days. As they age, they become less flexible and are either ruptured or phagocytized in the spleen. The breakdown of red blood cells is a carefully regulated process that ensures the recycling of valuable components. The globin portion of hemoglobin is broken down into amino acids, which are reused to synthesize new proteins. Meanwhile, iron from heme groups is salvaged and transported by transferrin to the bone marrow, where it is incorporated into new hemoglobin molecules. This recycling process helps maintain adequate iron levels for continuous red blood cell production. The remnants of heme groups that are not reused are converted into bilirubin, a yellow pigment that is transported to the liver and excreted as bile. Bilirubin metabolites are also eliminated through urine. If bilirubin levels become excessively high, a condition known as jaundice can develop, leading to yellowing of the skin and eyes. Disruptions in red blood cell function or production can lead to anemia, a condition characterized by reduced oxygen transport. Anemia is commonly assessed by measuring hematocrit and hemoglobin levels. Hemolytic anemia occurs when red blood cells break down prematurely, as seen in hereditary spherocytosis or sickle cell disease. Iron deficiency anemia results from insufficient hemoglobin production due to low iron levels. Understanding the life cycle of red blood cells and their disorders provides insight into how the body maintains oxygen balance and adapts to physiological demands. Iron is an essential component of hemoglobin, the oxygen-carrying protein in red blood cells. The body carefully regulates iron absorption, transport, and storage to ensure proper red blood cell production while preventing toxicity. Dietary iron is absorbed in the small intestine through active transport and enters the bloodstream. Once in circulation, iron is bound to transferrin, a transport protein that carries it to various tissues, including the bone marrow. In the bone marrow, iron is incorporated into heme, a key part of hemoglobin, allowing for the synthesis of new red blood cells. Red blood cells circulate for approximately 120 days before they are broken down in the spleen. During this process, hemoglobin is degraded, and the heme portion is converted into bilirubin. The liver plays a crucial role in metabolizing bilirubin and excreting it into bile, which is ultimately eliminated through feces. Some bilirubin and its metabolites are also filtered by the kidneys and excreted in urine. The liver also serves as a storage site for excess iron, which is stored as ferritin. This stored iron can be mobilized when needed to support red blood cell production, ensuring a continuous supply of hemoglobin. Through this tightly regulated system, the body maintains iron balance, supports oxygen transport, and prevents the accumulation of excess iron, which could be harmful. Platelets, also known as thrombocytes, are small but vital components of the blood. Unlike red and white blood cells, platelets are not full cells, but rather cell fragments derived from large precursor cells called megakaryocytes. These tiny structures are significantly smaller than red blood cells and contain membrane-bound vesicles filled with cytokines and growth factors. These contents play an essential role in cellular signaling, particularly in inflammation and tissue repair. Platelets have a relatively short lifespan of approximately 10 days, after which they are removed from circulation and replaced by newly produced platelets from the bone marrow. Their primary function is to prevent blood loss by initiating the clotting process. When blood vessels are damaged, platelets quickly adhere to the site of injury, forming a temporary plug. They also release signaling molecules that activate clotting factors, stabilizing the clot and preventing excessive bleeding. Beyond clot formation, platelets also contribute to immune responses and inflammation, interacting with white blood cells and other components of the immune system. Their role in both hemostasis and immune function highlights their importance in maintaining vascular integrity and overall health. Platelets, or thrombocytes, originate from large bone marrow cells called megakaryocytes. These giant cells have multiple copies of DNA within their nucleus and play a critical role in platelet production. Rather than dividing like other cells, megakaryocytes extend their cytoplasmic edges into blood vessels, where small fragments break off, forming platelets. In their inactive state, platelets appear as small, smooth, disc-like structures. However, when activated by injury or vascular damage, they undergo a dramatic transformation. Activated platelets develop a spiky outer surface and become highly adhesive, allowing them to stick to each other and to the damaged blood vessel walls. This response is essential for forming a clot and preventing excessive blood loss. Platelets are not only crucial for hemostasis, but also contribute to immune function and inflammation. They contain vesicles filled with cytokines and growth factors that help regulate wound healing and tissue repair. By understanding the relationship between megakaryocytes and platelets, we gain insight into how the body maintains a delicate balance between clot formation and normal circulation, ensuring both injury response and overall vascular health. Hemostasis is the body's natural process of preventing blood loss from damaged blood vessels. This essential mechanism involves three major steps: vasoconstriction, platelet plug formation, and coagulation. The first step, vasoconstriction, occurs immediately after a blood vessel is injured. The damaged endothelium and activated platelets release paracrine signals, causing the blood vessel to constrict. This narrowing of the vessel reduces blood flow to the affected area, minimizing blood loss. Next, platelet plug formation begins. Platelets adhere to the exposed collagen of the damaged blood vessel wall. Once attached, they become activated, swelling and changing shape to become stickier. Activated platelets release signaling molecules called paracrines, which reinforce vasoconstriction and promote further platelet aggregation. This forms a temporary plug that helps seal the injury. The final step is coagulation, or clot formation, where a cascade of clotting factors leads to the conversion of fibrinogen into fibrin. These fibrin strands create a stable mesh that reinforces the platelet plug, ensuring the wound remains sealed as tissue repair begins. Through these tightly regulated steps, hemostasis prevents excessive blood loss while maintaining normal circulation, playing a crucial role in wound healing and vascular integrity. When a blood vessel is intact, the endothelium releases prostacyclin and nitric oxide, which prevent platelet adhesion and keep blood flowing smoothly. However, when a blood vessel is damaged, hemostasis is activated to prevent excessive blood loss. The first step in platelet plug formation occurs when exposed collagen in the damaged blood vessel binds to circulating platelets, activating them. Once activated, platelets release signaling molecules known as platelet factors. These factors enhance platelet adhesion and attract additional platelets to the site of injury. As more platelets gather, they begin to aggregate, forming a temporary platelet plug. This plug acts as an initial barrier, helping to seal the damaged vessel and limit further bleeding. The platelet plug is reinforced by the coagulation cascade, which stabilizes the clot by converting fibrinogen into fibrin, forming a durable mesh. This process is a critical part of hemostasis, allowing the body to rapidly respond to injury while maintaining vascular integrity and circulation. Coagulation, or clot formation, is the final step in hemostasis, reinforcing the platelet plug with a strong protein network. This process involves the formation of fibrin, a tough, thread-like protein that stabilizes the clot and secures the damaged vessel. There are two main pathways that lead to fibrin formation: the intrinsic pathway and the extrinsic pathway. Both pathways are activated simultaneously in response to blood vessel damage. These pathways converge to activate thrombin, a key enzyme that converts fibrinogen into fibrin, forming a stable clot. However, clots are only a temporary fix. As the blood vessel wall heals, the clot must be removed through a process called fibrinolysis. Thrombin, in coordination with tissue plasminogen activator, or TPA, converts inactive plasminogen into plasmin. Plasmin then breaks down the fibrin mesh, allowing the clot to dissolve and restoring normal blood flow. This tightly regulated process ensures that clots form when needed to stop bleeding, but are later removed to prevent unwanted blockages, maintaining vascular health and circulation. The coagulation cascade is a series of biochemical reactions that lead to the formation of a stable blood clot. This process is essential for preventing excessive blood loss following an injury and involves two main pathways: the intrinsic pathway and the extrinsic pathway. The intrinsic pathway is triggered by damage to the blood vessel itself. It begins when collagen or other activators stimulate clotting factor XII, which then activates a chain reaction involving factors Xi, IX, and A. This pathway requires calcium ions and phospholipids to proceed. The extrinsic pathway, on the other hand, is initiated when tissue damage exposes tissue factor, also known as factor 3. Tissue factor interacts with factor 7 to rapidly activate factor X, providing a shortcut to clot formation. Both pathways converge at the common pathway, where factor X, with the help of calcium and phospholipids, converts prothrombin into thrombin. Thrombin then plays a crucial role in converting fibrinogen into fibrin, forming a stable meshwork that solidifies the clot. Additionally, factor 13 strengthens the fibrin network, creating a cross-linked fibrin polymer that reinforces the clot structure. This complex but highly regulated cascade ensures that clot formation is both rapid and controlled, preventing unnecessary bleeding while avoiding excessive clotting that could lead to blockages in circulation. The process of blood clotting, or coagulation, is essential for preventing blood loss after an injury. However, once the vessel is repaired, the clot must be safely removed to restore normal circulation. This balance between clot formation and breakdown is achieved through coagulation and fibrinolysis. Coagulation begins when thrombin converts fibrinogen, a soluble plasma protein, into insoluble fibrin. These fibrin strands form a strong polymer that stabilizes the clot and traps red blood cells, creating a secure seal over the injury site. As the damaged blood vessel heals, the clot is gradually broken down through fibrinolysis. Plasminogen, an inactive precursor, is activated by tissue plasminogen activator, or TPA, to form plasmin. Plasmin is the key enzyme responsible for degrading fibrin, breaking the clot into small fibrin fragments that can be safely removed from circulation. This carefully regulated system ensures that blood clots form when needed, but are later dissolved to prevent blockages in blood flow. The balance between coagulation and fibrinolysis is crucial for maintaining healthy circulation and preventing excessive clotting or bleeding disorders. Hemophilia is a group of genetic disorders characterized by the inability to properly form blood clots due to a deficiency or defect in one of the clotting factors in the coagulation cascade. Without functional clotting factors, even minor injuries can result in prolonged bleeding, increasing the risk of severe complications. The most common form of hemophilia is hemophilia A, which is caused by a deficiency of clotting factor VIII. This disorder primarily affects males due to its inheritance pattern on the X chromosome. Since males have only one X chromosome, a defective gene results in the full expression of the disease, whereas females can be carriers without experiencing severe symptoms. Uncontrolled bleeding, particularly in internal organs and joints, can lead to severe pain, joint damage, and in extreme cases, life-threatening hemorrhages. Without proper medical intervention, excessive blood loss from even minor injuries can become fatal. Treatment for hemophilia often involves the administration of clotting factor concentrates or recombinant factor therapy to help restore normal clotting function. Advances in gene therapy are also being explored as potential long-term solutions for managing this condition. Understanding hemophilia highlights the importance of the coagulation cascade and the delicate balance required to maintain proper blood clotting and circulation. In this chapter, we explored the fundamental components and functions of blood, covering 5 key topics. First, we examined plasma and the cellular elements of blood, understanding how plasma serves as the transport medium while red blood cells, white blood cells, and platelets perform critical physiological roles. Next, we discussed blood cell production, or hematopoiesis, which takes place in the bone marrow. We learned how different signaling molecules regulate the formation of red and white blood cells, as well as platelets. We then took a closer look at red blood cells, highlighting their biconcave shape, oxygen transport function, and life cycle. We also explored the essential role of hemoglobin and iron metabolism in maintaining red blood cell function. Following that, we examined platelets, the small cell fragments responsible for initiating clot formation. We explored their activation process and their contributions to wound healing and immune responses. Finally, we studied hemostasis and coagulation, the processes that prevent excessive blood loss after an injury. We reviewed the coagulation cascade, clot formation, and the breakdown of clots through fibrinolysis. Additionally, we discussed disorders like hemophilia, which affect the body's ability to clot properly. This chapter provided a comprehensive understanding of blood physiology, its critical role in maintaining homeostasis, and the mechanisms that protect the body from injury and disease.